The overall goal of this procedure is to determine forces on the linker of the nucleoskeleton-cytoskeleton, or LINC complex, through FRET microscopy in living cells via a biosensor designed for the LINC complex protein Nesprin 2G.
This method can help answer key questions in the field of mechanotransduction, such as the forces placed on proteins in the nucleus of living cells and how these forces change under varying conditions. The main advantage of this technique is its ability to measure very small levels of force on proteins in living cells.
Generally, individuals new to this method will struggle with capturing images. Optimizing the microscope laser power and gain to receive a good signal between all experimental groups is difficult.
To begin, grow NIH 3T3 fibroblast cells to between 70 and 90% confluence in a 6-well cell culture dish in a standard cell culture incubator with temperature and CO2 regulation. In a cell culture hood, remove the medium and rinse each well with approximately 1 milliliter of a reduced serum cell medium. Add 800 microliters of reduced serum cell medium to each well and place the 6-well chamber in the incubator. Then, pipette 700 microliters of reduced serum cell medium into a 1.5 milliliter tube labeled L with 35 microliters of lipid carrier solution to form the lipomix. Mix the suspension by pipetting. Next, pipette 100 microliters of the reduced serum cell medium into each of 6 1.5 milliliter tubes labeled 1 through 6. Pipette the plasmid DNA for Nesprin tension sensor, Nesprin headless zero force control, MTFP1, and Venus.